talking about? I'm a professor.
Where are all the pom-poms?
Yo.
Yeah.
Good. Because I'm cold.
officially. It's my great pleasure to introduce Michael Reed. He received his bachelor's degree in neuroscience from Northeastern University in 2011. He joined MIT in 2013 as a graduate student. And as a graduate student here at MIT, he received a number of different fellowships, including NSF Graduate Fellowship, Champions for the Brain, Whedon Fellowship, as well as Hofstad and Lisa Young Autism Center Fellowship. So essentially funded himself through, lucky for me. When he first joined my lab, he worked on a project that tries to look at the role of neuromodulators in olfactory associative learning. Let me just try to briefly dwell on this because I believe he's not going to talk about this in his presentation today. For this project, Michael, bravely proposed to test an existence of a neuromodulatory code in the piriform olfactory cortex that allows olfactory representations to be flexibly associated with multiple distinct types of unconditioned stimuli. In order to test this hypothesis or to search for such a code, Michael decided to teach himself a number of new techniques, which were non-existent in my lab at the time. And these included things like single-cell RNA-seq, CRISPR-based genome editing technologies, and he used these technologies to essentially screen through neuromodulators that are capable of imposing specific meaning on initially neutral representations of smell or odor in the piriform cortex. And at the end of this project, Michael was able to show indeed there is a neuromodulator called oxytocin, which is capable of endowing olfactory representation selectively with a social meaning in the piriform cortex. Subsequent to this project, Michael took another brave step, and in this next project, he asked whether molecules that are made by the immune system in the periphery, that's outside of the brain, can also work within the brain, similar to neuromodulators, to be able to change neural circuit function and thus to change animal behavior. And he asked this question in the context of trying to provide mechanistic understanding to this curious phenomenon whereby a subset of children with autism seems to somehow get better in their behavioral symptoms when they go through a course of fever, which usually accompanies systemic immune activation. Both of these questions that Michael has taken on as a graduate student, they were fascinating questions. They were fundamentally important and clinically relevant questions, but also at the same time, I have to tell you that these questions were complex and very much challenging. And I hope that you'll be able to see in his presentation today that the way Michael approached these questions reflected both his biological insights as well as his courage. You will also see the breadth and depth of his knowledge, his experience, his technical prowess that transcends the number of different fields, including immunology and neuroscience. And most importantly, I hope that by the end of this talk today, you will come to appreciate seriousness and rigor with which Michael conducted himself as he carried out his experiments and as he approached his scientific questions. So without any further delay, again, it's such a pleasure and privilege for me to introduce my first graduate student for his doctoral thesis defense seminar. Here's Mike.
Wow, thanks. So, like Gloria alluded to, this is not my complete work during my PhD, but largely the last 2 years of my experience in the lab. So I'm going to tell you about my talk, which is titled Fever-induced Rescue of MIA Behavioral Phenotypes. And this project is based largely on two things. The first one is, of course, autism spectrum disorder, which is a largely lifelong developmental disorder, which is characterized by delayed language or impaired communication, as well as social interaction, repetitive behavior, as well as restricted range of interest. And the second is this interesting phenomenon that largely circulates in anecdote form amongst the autism community, parents and doctors, which is that a subset of patients, when they develop a fever, autism patients, when they develop a fever, show a brief respite from their behavioral symptoms that last for the duration of the fever. And it was first reported in an autobiography of a parent of a child with autism in the 1970s, but I think that this quote from the Autism Speaks website sums up this phenomenon pretty nicely. She says, "Our 14-year-old has autism. We see striking improvement in his sociability, eye contact, organization, and mood when he's ill with a fever. It took a few occasions for us to make this connection. It's a clear, dramatic, but unfortunately temporary change." So this anecdote does not exist in isolation. It first was reported in the literature in this journal in kind of a Q&A format, in which parents are allowed to ask doctors a series of questions, and then the doctors will give them a reply. And the question they asked in this issue was, why do some autistic children seem to be nearer normal during episodes of high fever? And this is filled with all kinds of different anecdotes, including one in which clinicians at a psychiatric hospital at NYU noticed that some of their patients, when they developed a fever, also showed a relief of their behavioral symptoms. And there's lots more, including this very unscientific informal poll online in which they asked if your child improves when he has a fever, and roughly 70% said yes. And this wasn't grounded in the scientific literature. It wasn't systematically analyzed until 2007 in this pioneering pilot study of approximately 100 patients with autism, where the authors asked a very simple question, which was, do these patients actually improve during the course of a fever? And the experiment consists of measuring the patient's behavior at 3 different time points: when fever was detected, after fever was gone, and also a washout period approximately 1 week after the fever had dissipated. So they analyzed a subset of behaviors that are relevant for patients with autism, including irritability, which showed a very mild improvement during fever. Also, there was a large increase in lethargy, which you would expect given that the patients are sick, but also a pronounced reduction in hyperactivity, stereotypy, and inappropriate speech, which are hallmark symptoms of autism, but only a subset of them. So a follow-up study showed that approximately, or a much larger follow-up study of approximately 2,000 patients showed that approximately 17% of patients with autism are reported to show an improvement in their behavior during fever. And importantly, this study extended the findings of the first and showed that of patients that are reported to show an improvement, they show improvements in communication as well as social interaction. So these data suggest that a subset of patients improve, and that these can affect at least seem to be able to affect at least all of the core behavioral deficits that are reported in patients with autism. But also the authors noted that the distinction between those that improve and those that don't improve seems to be correlated with the severity of their behavioral phenotypes. So the authors noted that more patients who show an improvement during fever are administered this particular type of diagnostic test for autism, which is administered to patients who are nonverbal. So this means that more patients who are improved tend to be nonverbal than those who improve. They also show lower nonverbal IQ, also more severe stereotype behavior, social responsiveness, and stereotypy, which is the same as this but picked up again by a different assay. Also hyperactivity and inappropriate speech. So all of these studies suggest that the fever effect, which is largely reported anecdotally, may in fact be real. And we wanted to know, of course, why is this happening? What's the mechanism that could be producing this fever effect? And we chose to model this in mice, largely because of the experimental access that you're afforded in the mouse model. We chose to use the MIA model of neurodevelopmental disorders. This model is based on the observation that inflammation during pregnancy or infection during pregnancy increases the probability of autism in offspring. So either a bacterial or a viral infection during pregnancy increases the rate of autism and produces offspring with deficits in social behavior, communication, as well as repetitive behavior. And this is also can be mimicked in mice. So if you induce inflammation in mice during gestation, you can produce offspring that show deficits of social behavior, abnormalities in communication, as well as increased repetitive behavior. So in order to mimic fever, we chose to use lipopolysaccharide. So lipopolysaccharide is very commonly used in the literature in order to study fever. It is composed of a chain of carbohydrates that exists on the outer leaflet of bacteria. So this is composed of a chain of sugars as well as a fatty acid tail. And it's detected in the body by a toll-like receptor. And these receptors are very fundamental receptors of the innate immune system that are designed to detect the variety of pathogens that your body experiences. And following LPS binding TLR4, which is the type of toll-like receptor that binds LPS specifically, there's an induction of cytokines and interferons that are important for the initiation of the immune response during infection. And following infection or LPS administration, there's a number of changes, including fever, heat-seeking behavior, as well as lethargy and social avoidance, and also cellular and molecular changes that are fundamental to the immune response, like cytokine secretion, immune cell proliferation, as well as immune cell recruitment. So we wanted to know if we can model this in mice, and there's two things that are critical for this to occur. One is, can we induce fever in mice? And two is, when we induce fever in mice, do we see a behavioral improvement? So the first thing that we did was test whether LPS can produce a fever. And we did this by implanting mice with temperature loggers in their abdominal cavity. And then after waiting approximately 2 weeks for recovery, we injected these mice with LPS to induce immune stimulation, and then we measured their body temperature. And this is the temperature profile of control mice that have injected with LPS. You'll notice that there's a large spike in body temperature here. This is characteristic of picking up the mouse and injecting them. Their body temperature increases just from that experience. But you'll notice that the dark blue line, which is mice injected with LPS, show a slow creeping increase in body temperature. So this suggests that we can, in fact, induce a fever in mice. And of course, the most important question is, does this LPS administration and correlated fever produce a behavioral change? So this is kind of a behavioral setup that is very common in this paper, in which we inject LPS or give some kind of manipulation, and then we wait 4 hours. The reason we chose this 4-hour time point is because of the detection of the increase in body temperature occurs between 4 and 5 hours. And then we read out the animal's behavior. And this is a schematic showing the assay that we use most commonly in this paper, which is the 3-chambered sociability assay. And this assay is a very common behavioral assay in neuroscience, in which the animal is given a choice when exploring an arena to interact with an inanimate object or a social target. So generally mice that are wild-type control mice tend to interact mostly with the social target, and this is just a quick video showing kind of what this behavioral assay looks like. So the mouse starts on the inanimate side and then eventually goes over and explores the social target, and this is pretty representative of what happens during this assay. The mouse and the other mouse, the conspecific mouse, just kind of hang out and interact with each other. And this is these are two heat maps, or representative heat maps of the where the mice tend to spend their most time. So these PBS control mice, these are offspring that are derived from mothers who are injected with PBS or vehicle during pregnancy. And these mice are control mice, and they tend to spend most of their time hanging out with the social target, which is pretty typical. But MIA offspring, who are born from mothers who are injected with poly-IC, or when they have their immune system activated during pregnancy, these mice seem to spend approximately equal time in investigating either the social target or the inanimate object. So this is somewhat parallel to the symptoms that you see in some autistic patients, which are have social deficits. So we wanted to know, of course, whether LPS can actually affect their behavior. So we injected these mice and then waited 4 hours and then read out their sociability using this assay. And you'll see this type of plot over and over again in this presentation. For those of you who are not in science, each dot represents an individual mouse. and then the bar graph represents the average. And we tend to do a pre-test, which is the day before we give any kind of manipulation. We measure their baseline behavior, and then the test session is 4 hours after some kind of manipulation. So PBS mice, which are control mice, they tend to spend about 70% of the time interacting with the social target when injected with a vehicle. But if we inject them with LPS, there's also still no change. And MIA offspring tend to spend about 50% of the time interacting with the social target, which is a reflection of their sociability deficits. But if we inject them with LPS, although they have an asocial phenotype during the pretest, following LPS administration, they show a recovery to approximately wild-type levels. So we think that LPS can induce a fever. It can also, in fact, change their behavior. So this suggests that we might have a model in which we can study this fever effect that is observed in humans. And also, the human studies show that the behavioral consequences of fever are correlated with the temperature change. So when patients experience a fever, their behavior improves, but after the fever dissipates, the effect is gone. And we also see similar results that 3 days following LPS administration, MIA offspring that are injected with LPS show approximately 50% sociability. So we think that, much like is observed in humans, this is a transient effect. And we wanted to make sure that this wasn't just an artifact of the three-chambered sociability assay, which is kind of an artificial assay of sociability. So we chose to measure their sociability using a different assay called the reciprocal social interaction test. It's a very simple assay where you just place two mice and put them in the same arena, and you let them sniff each other, interact, and you quantify how much time they spend sniffing each other or touching each other. So much like with the sociability assay or the three-chambered assay, PBS offspring tend to spend more time interacting with each other, whether they were injected with vehicle or whether they were injected with LPS. But MIA offspring that were injected with LPS show a recovery to approximately wild-type levels, and also importantly, show a deficit in the absence of immune stimulation. So this is parallel to our observation using the three-chambered assay and suggests that maybe, in fact, at least in mice, the sociability is affected by LPS. And importantly, we wanted to extend our findings to a different core phenotype of MIA offspring, which is their repetitive behavior. And we read this out using the marble burying assay, which is another common assay in the autism mouse model literature. It's an assay in which you place a mouse in a cage that's filled with bedding with marbles placed on top. And wild-type mice tend to spend very little time interacting with these marbles, but MIA mice, as well as other autism mutant mice, spend a lot of time burying these marbles. This is a proxy for repetitive behavior in mice. And so here's a quantification of that. PBS mice tend to spend very little time burying, and when they're injected with LPS, there's a non-significant decrease. And MIA offspring spend or bury slightly more marbles, and if we inject LPS, this goes down to approximately wild-type levels. So LPS can induce a fever, it can affect social behavior, and can also affect another core phenotype of MIA offspring. So all the data I've shown you so far is using this MIA model, which is modeling an environmental risk factor, which is immune stimulation or infection during pregnancy. But there's many other mouse models that exist in the literature. Most commonly, these are monogenic models of autism, which harbor mutations in autism risk genes and also show behavioral deficits, much like MIA offspring. So we wanted to know if our results in MIA offspring are generalizable to these other models. So we injected LPS into these mice and then read out their behavior. So wild-type offspring, like PBS offspring, spend most of their time interacting with the social target. And catnap and FMR1 and shank mice tend to spend, or are approximately asocial. And if we inject them with LPS, which is these bars here, we don't see much of a change. So the effect of LPS seems to be, at least amongst these models that we tested, seems to be unique to MIA offspring. So now that we've kind of established a model for the fever phenomenon, we want to know what could be producing this change. So the most obvious variable to test is the temperature change that occurs during fever. So we wanted to isolate temperature as a variable and modulate the animal's body temperature in the absence of systemic inflammation. And we took our cues from the thermoregulatory circuitry in the brain, which after infection, there's an increase in cytokines which cause this molecule here to bind to the subregion of the ventral hypothalamus. And this causes a cascade of signaling that ultimately results in changes in body temperature. And people have shown that particularly in this region of the ventral hypothalamus, there are thermosensitive neurons that respond to changes in body temperature. And also controlling the activity within this region can control the animal's body temperature. So we chose to modulate the animal's body temperature using DREADS. So this is a receptor that when its ligand is bound, in this case produces inhibition. And we targeted this receptor to the ventral part of the hypothalamus in VGAT cream mice. And this receptor is conjugated to a red reporter, which is why you can see it in red over here. And when we inject mice that are expressing this receptor with the ligand, which is called CNO, we see a sustained increase in body temperature, which people have reported before. And you can tell that it lasts for approximately 2 hours. It actually goes for a little bit longer. And around 2 hours is when we assess the animal's behavior. So 2 hours after injection of CNO, in these are importantly, these are MIA mice who show sociability deficits. When we inject vehicle, there's no change, and when we inject CNO, there's no change in their sociability. So this body temperature change, at least after about 2 hours of a 1 degree Celsius body temperature change, there is no detectable change in sociability. But importantly, if you remember, LPS can increase the body temperature of control mice, but we did the same experiment in MIA offspring, and we don't see a change in the animal's body temperature. So these experiments show that, at least in the conditions that we tested, the temperature increase is not sufficient for the behavior. And also, even though MIA mice show a behavioral change following LPS, they do not show a temperature change. So it's very likely that temperature is not the variable that is driving this behavioral change. And also, importantly, it shows that MIA mice respond fundamentally differently to immune activation. So we want to know what are the mechanisms that are producing the LPS rescue, if not temperature. So we know from previous work in the lab that this one subregion of the somatosensory cortex is very important for MIA behavioral phenotypes. So Youngshin in the lab, she found that this brain region in MIA offspring are characterized by the presence of these patches, as we call them, which are actually just an absence of a cortical layer marker. So these patches are found kind of all over the cortex, but they're preferentially found in the S1DZ. And the presence of these patches is also correlated with an increase in CFOS. So CFOS is used in neuroscience. It's a quick and dirty way of reading out neural activity. It's correlated with increased neural activity. So you can see that in PBS mice, in their S1DZ, it's very quiet, not very much CFOS, so not very much neural activity. But in MIA offspring, there's a lot of CFOS. So we think of this as a hyperactive. And this is quantified down here. The S1DZ shows more CFOS. Also the neighboring forelimb region of the somatosensory cortex. And importantly, we think that the activity in this region is essential for the manifestation of behavioral phenotypes in MIA mice. And we tested this by taking wild-type mice and injecting channel rhodopsin into their S1DZ. That's depicted here. So channel rhodopsin is a protein that when you shine blue light on it, it increases the activation of neurons. So the idea was to mimic the hyperactivity that we see in MIA offspring. And in fact, as you would expect, this increases CFOS in the S1DZ. And also remarkably, it increases their marble-burying phenotype, which is the assay for repetitive behavior that I showed you before. And also importantly, it rescues it induces MIA-like behavioral deficits. So increasing activity in the S1DZ of wild-type mice, which normally show normal social behavior, and also normal marble-bearing behavior, can induce MIA-like behavioral phenotypes. And also importantly, reducing this hyperactivity using halorhodopsin, which is a protein in which you shine yellow light on, can inhibit the activity of neurons, can rescue MIA behavioral phenotypes. So turning on yellow light during the marble burying task reduces the marble burying phenotype of MIA offspring to approximately control levels. And this is also true when we read this out using sociability. So this is what a normal control offspring looks like, and this is an MIA offspring in which inhibition is presented. So all of these experiments are to say that hyperactivation in the S1DZ is extremely important for the manifestation of behavioral phenotypes. And it's not just important for MIA. We also found that hyperactivity as measured by CFOS is also extremely important for these monogenic mice. So relative to wild-type mice, the catnaps, the FMR1s, and the shanks show more CFOS in the S1DZ. And also, importantly, the magnitude of CFOS increase is correlated with their sociability phenotypes. So in these plots over here, we have sociability, and on the bottom, this is CFOS induction. And you can see that the more CFOS, the lower the sociability score. So this means that the hyperactivation in the S1DZ is correlated with the severity of the behavioral phenotype in catnap mice and as well as FMR1 mice. But it is only weakly correlated in shank mice to a non-significant extent. And we did the same experiment that I mentioned to you before, in which we inhibit activity in this region. We're trying to lower the hyperactivity in the S1DZ of these monogenic mice. So we shined yellow light into the cortex during the sociability assay. And this does not affect wild-type mice. They tend to spend around 70 to 80% of their time interacting with the social target regardless of inhibition. But in catnap and FMR1 mice, you'll notice in these yellow bars, which is when yellow light is being presented to these mice, you see a transient increase in their sociability. And this suggests that the inhibition in these mice can rescue their behavioral phenotypes. So this is all just to say that these mice also show hyperactivity in the S1DZ, and this hyperactivity is functionally tied to their behavioral phenotypes. So given how important hyperactivity is for MIA behavior and monogenic behavior, we wanted to test whether the behavioral rescue that we see following LPS is mediated by a similar mechanism. Does LPS mediate its behavioral rescue by reducing the hyperactivity in the S1DZ? So we did a simple experiment in which we injected mice with LPS, and then 5 hours later, we sacked the mice and then read out CFOS in their S1DZ. So here's some histology showing CFOS in the S1DZ. PBS mice tend to not really show much CFOS in the S1DZ. It's very quiet, like we showed before, and MIA mice show relatively more CFOS. But if we treat them with LPS, this seems to go down to wild-type levels. And this is not a brain-wide phenomenon. We measured several other cortical regions that I'm not presenting here, and this seems to be somewhat unique to the S1DZ, although there is a almost significant trend in the barrel field, which is immediately lateral to the S1DZ. And also importantly, we chose the central amygdala as kind of a control region because people have shown in the past that this region responds to aversive stimuli, in particular LPS administration. And even though we don't see much of a change in LPS in the S1DZ here in PBS mice, and we don't really see any CFOS in the S1DZ following LPS in MIA mice, we see a lot of CFOS in the central amygdala. So this is all quantified here. MIA offspring show more CFOS, and this is reduced upon LPS. And also, LPS administration in either control mice or MIA mice strongly induces CFOS in the amygdala. Now, if you remember, MIA offspring are the only ones that show a behavioral rescue following LPS. The monogenic mice don't show any behavioral change. So in order to ask whether this reduction in neural activity could in fact be tied to the behavior, we wanted to see if this also happens in the monogenic mice. Because if it does, then that suggests that it's probably not that important. So we did the same experiment where we injected LPS and then read out CFOS. And you'll notice that there's a lot of green dots here suggesting mirroring the CFOS induction that I showed before. These mice are also, like MIA mice, hyperactive in the S1DZ. But injecting LPS doesn't really seem to change very much. So you can kind of tell that in the example histology images, and it's also very clear in the quantification. But the amygdala still reliably responds to LPS, suggesting that these mice are not impervious to the effects of LPS. So why is LPS uniquely affecting MIA offspring? We got some clues from some previous work showing that the immune system of MIA offspring is fundamentally changed into adulthood. So if you remember, we inject these mice with an immune stimulant during pregnancy, or the mothers of these offspring with an immune stimulant during pregnancy, and it's the offspring that show the behavioral phenotypes. And T cells derived from these adult offspring seem to be more sensitive to secrete cytokines in response to stimulation. So they show relatively elevated levels of IL-6 and also IL-17, and also, but don't show a change in TNF-alpha, and this is stimulation in culture. So we wanted to do the same experiment, but in mice. So we injected LPS into these mice and then read out a couple of different cytokines in the plasma. And we found that MIA offspring in response to LPS stimulation seemed to secrete more cytokines, with the exception of TNF-alpha, which is consistent with the results that I just showed you, but they show more interferon gamma and also IL-6 and also IL-17 induction in response to LPS administration. And we did the same thing in the monogenic mice. But importantly, IL-17 seems to distinguish MIA offspring much better than the other cytokines. So the monogenic mutant mice also don't really show, or the monogenic mutant mice do not show induction of IL-17 following LPS administration. So we thought that IL-17 might be a good candidate cytokine mediating the effects of LPS on the behavior of MIA mice, and could possibly explain why we see no effect in the monogenic mice, but a strong effect in MIA mice. But in order for this to be true, the receptor needs to be expressed in the brain. So in order to test this, we did an in situ hybridization for the receptor that binds IL-17A. So we found that the receptor for IL-17A is strongly expressed in the neocortex. It is also expressed in the basolateral amygdala and also expressed in the hippocampus. So it seems to be the case that the IL-17 receptor is preferentially expressed in cortical regions. And we validated that this signal is real in IL-17 receptor knockout tissue. So what you might not be able to appreciate from that kind of zoomed out image is that the receptor is actually expressed in the entire cortical depth of the cortex. It is, on a per cell basis, much more highly expressed in layer 6, but also expressed in layer 5. But it is also represented in layers 2, 3, and 4. So these data suggest that IL-17 has the ability to affect the brain. And importantly, we wanted to know if this can affect neurons. So we did a co-staining of IL-17 receptor as well as NuN. So NuN is here in green. NuN's a protein marker that marks neurons. So we looked at co-labeling of IL-17 receptor in NuN positive cells in the cortex, and we found that 90% of neurons in the S1DZ express the IL-17 receptor. So IL-17 at least has the ability, whether it does or not is unclear at this point, to affect neural activity in the brain. But we wanted to know that if it's actually doing anything. So we did an experiment in which we implanted mice with a cannula into the S1DZ, or right above the S1DZ, and we injected recombinant IL-17A directly into this brain region bilaterally, and then we measured out their sociability. And we found that in PBS mice, IL-17A injection doesn't really affect their behavior. They still show their characteristic approximately 70 to 80% sociability score. And MIA mice injected with vehicle into the brain show their characteristic of approximately 50% phenotype. But mice that are MIA mice that are injected with IL-17A directly into the S1DZ show a recovery of their sociability phenotypes to approximately wild type levels. So IL-17A at least is sufficient to promote sociability in the brain, and possibly through the receptor that I showed you before. But interestingly, this is also true for at least 2 of the monogenic mutant mice that I showed you. So we didn't pursue the Shank mice any further because they didn't show an effect with inhibition and did not have correlated phenotypes with S1DZ CFOS. But when we injected IL-17A into the S1DZ of these monogenic mice, just like we did with the MIA mice, we see a promotion of their sociability. So these mice although they don't show IL-17A induction following LPS, if we bypass that by directly injecting it into the brain, we can see a promotion of their sociability. So the effect of IL-17A seems to be somewhat generalizable, or at least is not unique to MIA offspring. So then we want to know if IL-17A is actually important for the effects of LPS. So the sufficiency experiment shows that it at least can do something in the brain. It can promote behavior, but does not necessarily demonstrate that it's important for the effects that we see with LPS. So in order to ask this question, we pre-treated mice prior to LPS injection with a blocking antibody targeted against IL-17A. So this prevents the ability of IL-17A to interact with its receptor. And here's a PBS offspring injected with vehicle or LPS. There's no effect like I've shown you before. MIA mice injected with LPS but with a control antibody in their brain show the characteristic rescue. So this is effectively just measuring the effect of LPS, which I've shown you before. But if we pre-treat them with blocking antibody against IL-17A, blocking the effects of IL-17A in the brain, we see prevention of the effects of LPS. And this is at least somewhat unique to IL-17A because blocking interferon gamma signaling in the brain, which is another cytokine that's upregulated upon LPS administration, doesn't seem to affect doesn't seem to prevent the effects of LPS. So IL-17A is important for the effects of LPS on behavior, but we also know that LPS can reduce the hyperactivity in the S1DZ. So we wanted to know if IL-17A is actually mediating this reduction or mediating the effects of LPS on neural activity in the brain. So we did a similar experiment to what I showed you before, in which we read out CFOS induction in the S1DZ. So it's a way of reading out neural activity. But we pre-treated them with blocking antibody against IL-17A. So this graph here is showing the difference between PBS and MIA offspring. So like before, mice MIA offspring show increased CFOS induction in the S1DZ. But if we inject them with LPS, this goes down like before. But if we pre-treat them with anti-IL-17A, this reduction in CFOS is prevented. But this does not happen when we look at our control region, the central amygdala. If we inject LPS, regardless of whether we pre-treat it with a blocking antibody, we see strong CFOS induction. So it's not preventing the effects of LPS brain-wide. But all of the data that I've shown you so far on neural activity is using CFOS, which is very commonly used in neuroscience, but it's kind of like a quick and dirty experiment that people do just because it's easy. But we wanted to know if these effects were real, so we implanted mice with electrodes in the S1DZ in order to directly read out their neural activity. So this was done in collaboration with Michael Halas's lab, primarily driven by this excellent research scientist, Ralph Wimmer. So we recorded activity in the S1DZ while these mice were walking on a wheel. And prior to, or we would establish a baseline, and then following baseline, we'd inject these mice with a blocking antibody in order to assess the necessity of IL-17A, and then following LPS. So we found that this experiment here, this is example raster plots from the experiment in which we pre-treated mice with a control antibody and then injected them with LPS. This is effectively just reading out the effect of LPS on IL-17 on S1DZ neural activity. So PBS offspring show no change between baseline and also 4 hours following LPS administration. So this suggests that, like we see with CFOS, there's no change in neural activity after LPS injection. But MIA offspring show a large reduction in their neural activity in the S1DZ. So like we saw with CFOS, this is exactly the same. We see a reduction in neural activity following LPS. And this is quantified over here in these box plots. If we treat PBS mice with LPS, there's no change. But if we pre-treat them with blocking antibody and IL-17 and LPS, we don't see a change. But MIA offspring show a large reduction in firing rate after LPS injection. And this is blocked by preventing IL-17A signaling. So it seems like IL-17A is capable of affecting neural activity in the brain and maybe mediating the effects of LPS. But all of the experiments that I've showed you so far are kind of leading to the point that IL-17A is affecting the S1DZ following LPS administration. But to ask these two questions, to really nail down whether the S1DZ is important and whether IL-17A signaling is important in the S1DZ, we knocked out IL-17 receptor expression bilaterally in the S1DZ. We made MIA offspring in the IL-17 receptor A conditional knockout line, and then injected them in the S1DZ with either something that just expresses a reporter or CRE, which is mediating the knockout. So all of these cells in this picture, for example, would be expressing CRE and therefore have IL-17 receptor knocked out. And then we did the experiment in which we injected these mice with LPS and then see if we see the behavioral change. So these mice are control mice that are just expressing reporter in the S1DC. And if we inject them with LPS, we see the characteristic rescue of their sociability phenotypes. But if we knock out IL-17 receptor expression in the S1DZ bilaterally, LPS is no longer able to produce the rescue. So these data suggest that not only is IL-17 important, it's especially important in the S1DZ for this rescue. So in summary, what I've showed you is that LPS can affect behavior, both sociability as well as the marble-bearing phenotypes. Also, LPS or IL-17A induction in MIA offspring distinguishes MIA offspring from monogenic mice, and therefore establishes IL-17A as a candidate for mediating the effects of LPS. LPS reduces hyperactivity in the S1DZ, as both measured from CFOS induction as well as single unit recordings in the S1DZ. And also, receptor expression in the S1DZ is critical for the effects of LPS. So I have certainly a lot of people to thank for this. Members of the Choi Lab, Gloria, Youngshin and I worked very closely together on this project and also on many different projects throughout the years. Also Han was the first postdoc in the lab who was there when I was rotating. He was a large reason why I joined the lab in the first place. He's a very charismatic but good scientist and was also working on very cool stuff. Also Matthias, who was essential for this project. Also Chanyang, who did some electrophysiology data that I'm not showing here. And also Natalie, who kind of functions as like the glue of the lab and has been supporting me and Youngshin in our research pursuits for the past 6 years. And also former members of the Choi Lab, Ashley Park and Nora and Lila, who have since become great friends. And also the Alec and Dan and Hunter and Juntae and Byungjin and Liu, who are also members of the Choi Lab. I also have to thank members of the Ho Lab, in particular June, who's been like a second mentor over the years. Also Gwyn from the Sci Lab, who worked very hard on a bunch of RNA sequencing experiments that I'm not showing here. Also the Halassa Lab, particular Ralph, who spearheaded the electrophysiology experiments. And also Melissa and Yang, who supported us through this project. And also Matthias from the Zhang Lab, I'm not showing this data here, but it's related to the experiments that Gloria was talking about in CRISPR and whatnot. He was very generous with his time in teaching me how to do some basic molecular biology and also just generally how to use CRISPR, which is, at the time, I remember in my first meeting with him, not understanding anything he said, and then just through his patience, being amazed that I was able to do tissue culture and make virus and do all these other things that were very amazing to me at the time. Also, I have to thank my committee, June, and also Mraganka, who is credited with promoting the electrophysiology work in this lab. That's been his primary comment during my committee meetings over the years. And also Troy, who is also credited with the expansion of our behavioral experiments beyond just sociability. Also, he probably doesn't remember this at all, but during my first year, we went to like a recruitment party, and he very strongly urged me to join Gloria's lab because I would have the best project in the lab because it was a new lab. So he was a strong facilitator for me ultimately joining the Choi Lab. And I'm very grateful for their input and wisdom over the years and guiding me in my project. Also have to thank my funding, McGovern, as well as the Hawk Tan and Lisa Yang Center for Autism, and also the NSF. And I also have to thank, in particular, Kay Tai and Kai Goosens. Kay Tai, the work that I talked about in the NSF was from the TI Lab, and she was very helpful in crafting that proposal, as well as members of the TI Lab. And also at the time, there was an NSF workshop that was led by Kai Goosens that led to my entire class getting the NSF, which was pretty amazing, and she was a huge factor in that happening. I also want to thank my science family. So these two guys over here, this is Jim Steller. I credit these guys with getting me interested in neuroscience in the first place. So Jim was the dean of the department, or dean of the School of Arts and Sciences at Northeastern, but he also had a lab. But there were no postdocs, there were no graduate students. It was just like 15 undergrads who had way more responsibility than undergrad should ever have. But those formative experiences were the key to me sticking with the neuroscience over the years. And I'm very grateful for his support over the years. Also, Marcelo, who I worked with at Northeastern University, he's like a big teddy bear of a guy, very charismatic, and also a great scientist, and like Jim, gave me a lot more responsibility than an undergrad should ever have. But I'm very grateful for the work that we were able to do together, and I was very privileged to be able to work with these two at Northeastern. Also, June I think has functioned as like a immunology encyclopedia over the years, and is kind of like a great person to gut check your experiments with, because he seems to know everything. Also, I'm grateful for his positive attitude during our weekly meetings, which made them both fun and productive. Also, of course, Gloria, being the first graduate student in the lab, I'm extremely grateful that she was willing to take a chance on me. As I've heard people describe that joining an assistant PI's lab, particularly as their first graduate student, is like betting on a horse. You never know what's going to happen, which is also true for the first graduate student. And I think that this bet paid off pretty well for the both of us. And my experience in the lab, I think, has fundamentally changed me as a person and a scientist. And I think I would have never imagined where I am right now years ago, and certainly not where I'm taking my career next. And I credit that to Gloria and also my experience in the lab. So thank you very much. Also, I want to thank my classmates. This is only a subset of them, but also in particular Chris and Dennis, who have become great friends to me over the years and have been very supportive during the dark times, the many dark times of grad school, and also the good times. And I don't blame them for all leaving before I did. Also, this is a snapshot of the Choi Lab over the years. So this is Daniel back when he was a tech in the lab, who's here today. Also Han, who was the first postdoc in the lab, and Nora, who came with Gloria from Columbia to establish the lab. I'm very grateful for Nora during my first years in the lab. And also this is Youngshin shortly after she joined the lab. Then the lab evolved and added more postdocs, and John Young as well. And also I have to thank the people who have been with me for the 25 or so years that I was not a graduate student. So this is my sister and my brother from a Vermont State Fair, I think, eating some candy apples. And here we are today, and now my sister has 2 kids. And of course, my parents, for accepting my lack of availability pretty much over the past 6 years and being very patient and supportive. And also my grandparents, who really wish I went to Duke, but it's okay. Fortunately, Duke didn't let me in, so they made a very easy decision. They are a wonderful supportive influence, and my grandmother actually got the chance to visit the lab when I was a first-year graduate student, and just persistently asked me one question the whole time, which was, how does this relate to Alzheimer's? I was like, it's like, not at all. And that was when I was doing olfaction stuff, which is totally not related to what I'm doing right now. Also, Jan, Nora, and Ellen, who at the time in the early days were all technicians in the department and have now since become graduate students on their own. I'm very grateful for their friendship over the years. Also, Dave, who's here today, has been a great friend. We met in the Jim Steller's lab back at Northeastern. He was my de facto supervisor, although he was only like 3 years older than me and also an undergrad. But somehow we were able to get our stuff together and actually publish the stuff that we worked on, even though it's only one figure, that's fine. But yeah, Dave has become a great friend and also I think functions as like a scientific older brother, which has been enormously helpful over the years. Also, Youngshin, who's joined the lab about 6 months after I did, who I think any perceived success that I've had over the years, you can pretty much attribute to Youngshin's contribution. She's extremely good scientist and also a very patient and caring person. So she, in addition to Han, or mostly Youngshin, but Han definitely got me started, taught me pretty much everything that I know in the lab and was extremely patient with teaching me everything, helping me with my experiments, staying late in the lab, coming in on the weekends. I definitely would not be here today, and certainly this paper that we've been working on would not be published right now if it wasn't for Youngshin's help. So I'm extremely grateful for Youngshin's involvement in my scientific and also personal life over the years. And she's, I think, effectively been integrated into my immediate family since she started. Also, I want to thank Colin, Justine, and Leslie. These are some of my friends from high school who we occasionally take fun trips together like this. And also Sarah, who's been a great partner over the years and also a wonderful resource for immunology since she has her PhD in immunology. But she's been very helpful and very supportive over the years, and I'm very grateful for her role in my life. Also, I need to thank the newest member of my family. This is Pickles II. Pickles is a 10-week-old French bulldog. He's definitely the smelliest member of my family. And if you can't see him, this is here. He's right here in these tiny little circles. Anyway, and also thank you guys for your attention. Well, it seems there's some RNA sequencing data that suggests that it's expressed in the pyramidal neurons preferentially. So, I mean, it's also expressed in inhibitory neurons. And also one important thing that I'm not showing here is that the receptor A is only one of the subunits of the complex. There's also receptor C, and we don't know exactly where that's expressed in the brain. So that might be dictating there might be more specific cell types that are expressing the complex than what we show here. But my expectation based on the sequencing data is that it's having an inhibitory effect on pyramidal neurons.
I'll ask one. What is it about the S1DZ?
I mean, the short answer is we have no idea. I mean, it's like involved in proprioception, but how proprioception could be mediating social behavior is pretty unclear.
So it seems like IL-17 receptor is everywhere else.
Yeah.
Roughly in cortex.
So the effects of...
Where you rescue it outside of S1DZ?
So we haven't done that with IL-17, but we have done that previously with the optogenetics in Youngshin's paper. So if you can't induce MIA phenotypes in regions outside of the S1DZ. So it seems to be somewhat specific. There's something special about S1DZ, but I definitely don't think the effects of IL-17A are specific to S1DZ. I just think its effects on the S1DZ produce the rescue. And just the effects elsewhere are not mediating, or something socially relevant, I guess.
So thank you for coming. We have after the event party, probably starting around 5 p.m. if you guys can stick around on the 6th floor. I think that's all the other announcements.
was okay? Good.